Hello, my name is Mr. Chipman. I teach biology and AP Biology at Murray High School in Murray, Kentucky. This is Unit 2, Cells 2.3, Plasma Membrane. Looking at the plasma membrane and the past that causa doom. Probably won't be looking at the second one, though that would be fun. We're just going to look at plasma membrane today. And here's a picture of the plasma membrane. A lot of times you'll see the plasma membrane the term for the plasma membrane is often used is the fluid mosaic model. So let's break it down using all three of these terms. First of all, it's a model. A model is the way that we suppose things behave or look, right? It's sort of a way to look at something in real time, or not in real time, but in kind of frozen time. It's a good way of looking at things, right? Understanding them. A mosaic, then, is something that's composed of multiple types of things, right? Sort of like if you think of the word being used in art, we use the mosaic when it's got lots of stuff going on. Well, if you look here, there's lots of stuff going on. And it's fluid. It's not static, meaning it's dynamic. It's moving around, right? And so all of these pieces, these proteins and these phospholipids and these squiggles, squiggles is not a technical term, they're all moving around. They're all constantly moving, and they're constantly moving in order to adapt to the cell's needs. So we're going to talk about some of these individual portions. Let's do that. One thing before we move on is the purpose of the plasma membrane is twofold. It allows for selective permeability and compartmentalization. Obviously, compartmentalization on a grand scale, keeping the cell separate from its environment. And then semi-permeability is the idea that it only allows certain things in and out, both in and out. Let's talk about phospholipids. We've talked about phospholipids already, but we're going to get into a little bit more here. So phospholipids have a hydrophilic head. That means that it's polar, right? And I like this picture because it kind of breaks down the components of that. You have this phosphate area here. You have a glycerol. Remember, glycerol is sort of the things that connects fatty acids together. This is a polar, very polar region here, likes water. And then you have two hydrophobic tails. These are fatty acid. One of them is saturated, the other one's unsaturated. And so when phospholipids get together, they will orient themselves in such a way that the hydrophilic portions want to be situated toward water, and the hydrophobic portions, or the nonpolar portions, want to be situated away from water, and they will form this sort of phospholipid bilayer, is the term that is often used here. Another word that you'll see used for phospholipids is the term amphipathic. And I'm going to just go ahead and type the term amphipathic here. I need to find it on my notes because spelling is not my strong suit. Amphipathic, right? And this is the term that means that they are both hydrophilic and hydrophobic. Now, not any one part is both, but it has two different sections. One of them is hydrophilic, the other one is hydrophobic. There are other amphipathic things that we're going to look at, but this sort of chemistry, the way this amphipathic nature of phospholipids allow the membrane to be semi-permeable. Certain things can come in and out, certain things can't, right? That brings us to membrane proteins. So if a protein is in a membrane, that protein must also be amphipathic, right? Certain portions of the protein need to be able to interact with the polar portions of the phospholipids, and the other portions need to be able to interact with the nonpolar portions of the phospholipid. And so membrane proteins come in two different ways. You have these integral proteins, which are called that because they go all the way through the plasma membrane. These two little gray lines are supposed to represent that bilayer. See how these proteins go all the way through. And a peripheral protein is on the outside. It's kind of like your peripheral vision, right? It's on the outside, right? You can't see it. It comes into your periphery. It Get the idea. Integral all the way through. Periphery or peripheral proteins are on the outside, either on the inside of the cell or the outside of the cell. And they sort of serve different functions. I think we're going to look at the functions. Yeah, we're going to look at the functions in this slide. They can function as transport. We're going to look at that in the next section, dealing with cell transport or protein transport. They can be used as signal transduction, which is where they'll receive, like act as a receptor for a molecule. And then we're going to look at the idea of enzymes as well. They can be used to anchor, this anchorage idea. They can be used to like connect cells together. There's a lot of different things that membrane proteins can be used for. Cholesterol. Cholesterol is not a membrane protein, but it is found in cell membranes. It is a lipid, right? You see the lipid structure here, the long chain, there's the lipid, and then a steroid always has these 4 ring structures. Cholesterol's purpose is to stabilize membrane fluidity. It's going to reduce movement at high temperatures and prevents packing, prevents instability at low, or stability, I guess is a better way to think about it, at lower temperatures. So it gives the cell membrane fluidity, and it maintains a normal level of fluidity at extreme temperatures. So it's pretty important. Cholesterol has a very important role in cell membrane chemistry or plasma membrane chemistry. Glycoproteins and glycolipids. So we see this prefix glyco. Anytime you see that it has to do with a sugar or a carbohydrate. A glycoprotein is when a carbohydrate is attached to a protein, and a glycolipid is when it's attached to a lipid. These molecules are very important in cellular recognition and signaling. And we've talked about glycoproteins and glycolipids a little bit, particularly glycoproteins when we looked at the function of the Golgi body or the Golgi complex. And so very important in cellular chemistry as well. Some other examples that you'll see of these membrane proteins in particular, MHC class 1 and 2, these are immune proteins. They're used for cell recognition. And so you can see how they're sort of anchored here in the plasma membrane, and then they have this area that's on the outside of the membrane that is going to be more polar. Some other things that we'll talk about when we get to transport are aquaporins. Think of the word aqua, water, porin, pour. It's literally a tube that allows water in. Sodium, potassium pumps. I'll give you one guess as to what they pump. But because it's called a pump, they do require energy. Well, again, we're going to talk about different kinds of transport tomorrow. Not tomorrow. Could be whatever day you're going to listen to it, but it'll be in 2.4. Let's look at a practice problem. In red blood cells, membrane proteins are embedded in a phospholipid bilayer that separates the cytoplasm from the external environment. Which feature of these proteins allows them to remain stably positioned, good wording, within the membrane? Okay, pause the video, take a second. All right, so what allows these proteins to be in the membrane, positioned there, right? It has to do with the fact that the proteins are going to be similarly chemically aligned as the membrane, right? Proteins are linked to ribosomes. No. Proteins are surrounded by DNA that anchors them in place. No. Why? What? Proteins have hydrophobic regions that interact with the interior and hydrophilic regions that face the aqueous environments. Of course, that's the answer. Let's look at D. Proteins are fully hydrophobic or hydrophilic. No, that's it. Again, we're not good there. We like answer C. Hopefully this was helpful. 2.4 coming soon.